Thanks, Vivash. So thanks for inviting me, and I changed the talk a little bit. Like, the title that is presented doesn't have the non-inferiority, but I thought that it will be much more useful, informative if I say, like, I'll be talking about SMART in the non-inferiority context. And again, this is a joint work with Vivash and my former postdoc, Irina, and Alla Sikorsky, who gave the real example, like, which motivated the study. And I should tell you, frankly speaking, I'm not a smart guy in that sense. So I'm really a randomized control trial guy. So I think Vivash visited Michigan maybe 2 years back. Yeah. So I think Susan was still in Michigan then, right? Before moving to Harvard. So as we talked, and our university is like about 50 miles away from the Ann Arbor. So when he visited me, like, we were talking, and then we are discussing, like, what you do, what I do. So I do that standard clinical trial. So and non-inferiority is a special interest of mine for several years. And Nayo is here, like, who supported me for a long time, non-inferiority trial design and stuff like that. So then we are talking, okay, can we do something non-inferiority in the SMART trial? So then he was very excited and told me, I have a almost done paper in non-inferiority trial in SMART. So then I asked him what kind of non-inferiority you're doing, because there are different variety of non-inferiority. Now, of course, he's not a non-inferiority guy. Just like I'm not a smart guy, he's a non-inferiority guy. So, so then I discussed, like, do you have done this kind of non-inferiority? Do you have done other kinds? So, no, we have done a special kind. So you have much more to do. So this particular talk came out of that collaborative work, and it has really two parts. Like, one part is non-inferiority, the other part is the SMART, and then you'll see engagement of both. Now, I should tell you that when I talk non-inferiority to a statistics department or mathematical science department, they say it's all done. Why? Because you just change the inequality and you are done, right? Everything is all done. It's not all done. When you go to a biostatistician and you try to implement it, actually there are much more price you have to pay when you just change the inequality, and you'll see some of it today. So let's see. So before I begin, as from a school of medicine, like, we don't do any research without money. So I should be thankful to my funder. So this is the outline of the talk, and again, like, as we are presenting here, it's not to finish the talk, the whole, but I will take question if you have any as we go. So I'll talk first about the clinical trial, and again, like, this is a very diverse audience. So again, many people have done clinical trial for long, so I'll talk a little bit about clinical trial, like maybe a couple of pages. And then I'll talk about special type of non-inferiority trial, like trial with non-inferiority trial, like, and there has to be a context. Why you will all of a sudden going for non-inferiority trial? There has to be a reason. Then we'll talk about SMART. Again, I'm pretty sure that a lot of people here know a lot more than SMART than what is presented here, but actually just to give you some introduction, like what is SMART. And then we'll talk about a special kind of SMART. Like, that is where, like, the novelty come here. We're talking about a 3-arm SMART. It's all about 2-arm SMART that we have seen so far, like, but can SMART be of 3-arm type? And that's where Allah's contribution come from Michigan State. He has an example where there is a 3-arm SMART, okay? And then I'll talk about simulation study and some conclusion and reference. And again, I know some of you are asking for the slides, but this is still not published. So I'm a little bit hesitant, but if you talk to me or ask me personally, I'll give you the slide. So what is efficacy seeking trial? Like, if you are a clinical trialist, clinical trial comes in different flavor, from efficacy to effectiveness to implementation. And each stage has its own challenge. They're all not all same. You cannot put an iron roller and everything is same. It's not same. So we will be talking about the efficacy seeking trial. If you go to comparative effectiveness research, you have to modify, you have to pay different price. So what is efficacy? Efficacy is de jure, which is the capacity of measuring the beneficial change of a therapeutic agent, which could be a drug. And I'm primarily a drug trialist, but it could be an intervention, it could be a devices, for example. Anything that you do to hinder the progress of the disease, that is an intervention. So efficacy, essentially try to measure that. So to measure efficacy, randomized control trial is used for last maybe more than 60 years. It's got matured, and we understand very well how to establish causality. Can you do this kind of experiment without randomization or randomized control trial? Yes. For example, people have done some work on observational study, But you have to pay some price because there is selection bias and causality principle is not so well established until unless you take care of that well. So RCT, and as Susan is not there, but Susan was explaining like design point of view, RCT is based on 3 basic principles of DOE, design of experiment. Like randomize, replicate, and blocking. Everything has a meaning, has a reason we do, okay? So you randomize because essentially you don't want to end up with a 0-1 kind of assignment design, which is a lot of selection bias. You replicate because you want to make sure that the effect that you're seeing is not a chance thing. And you block when you cannot essentially randomize, then you do stratification or the blocking. So RCT involves human subjects. So we'll be talking about not animal experiment, but we'll be talking about human subjects. And again, like, whenever you're using human subjects, you have to be very careful about the sample size. Like, we go to NIH review panel and standard thing is that I have large sample size. No, you cannot have large sample size. You cannot expose people for just to declare your drug is beneficial. There is an ethical challenge behind that. At the same time, you don't want a small sample size because then it's a failed trial. Okay, so that balance is very important because you are working with human subjects. Of course, when you work with animal, that's also interesting, but we'll be talking about human subjects. So we are talking about prospective control experiment under strict idealized condition. So this idealized condition makes this efficacy-seeking trial is different than effectiveness trial. Okay, because what you are observing is a very artificial environment that may not be exactly replicable if you go to a real world thing. So that's what, but I'm not talking about the real world thing. I'm talking about where things are much more idealized. So to declare something efficacious, we need a control group. Like it's efficacious compared to what? You have to have that thing. And the control group plays a very interesting story when you go to standard clinical trial to active control trial, which will come in a minute. Okay, so RCT, what is RCT? Going deeper, it's a randomized controlled clinical trial, and essentially it's a gold standard for determining whether something works, some drug or intervention works or doesn't work. It plays a huge emphasis on internal validity. And you will see in a minute that, like the 2-arm or the standard non-inferiority trial actually pay a huge price because they don't pay the price of internal validity. Okay, it's not internally, you cannot declare internal validity. The goal is to determine whether certain intervention works. And in the absence of an effective treatment, the usefulness of placebo-controlled RCT is uncontroversial. So sometimes we say placebo, sometimes we say standard of care. So when you have nothing and you are coming up with certain chemical agent or some certain intervention, it is ethically possibly okay to do an RCT with that kind of thing. However, ethics plays a very important role in clinical trial. So when you have an established agent or regime, then you cannot do an RCT with a standard of care or nothing, because you are putting patients in risk and without any benefit. Okay, and there is an agent already available in the market which will do a better job. So the question is then, what should be then the control group? Because you have to compare against something. Okay, so implication of placing an active comparator, that means you are comparing now a drug versus drug, or an intervention versus intervention, and one of them works, that, and you're trying to see whether my new intervention also works. So if you do that, you have to pay a heavy price. And now you can see, like, I can declare my trial of 2 different types. One is superiority trial, and another is a non-inferiority trial. Now, what is a superiority trial? Standard trials are superiority trials. You say that whether my drug or my new intervention is better than the existing one. And basically, that's what you do in standard clinical trial when you are declaring a new drug is better than the placebo. Okay. However, if superiority of the drug is in question, now of course you will not run a trial as a superiority hypothesis, and they say then, oh, okay, I can't beat it. So can I declare non-inferiority now? You can't do that in a single trial. Okay. So why the superiority will be in question? Well, before you go to the phase 2B or phase 3 trial, you get a lot of evidence. Okay, phase 2A, phase 1, you get a lot of evidence from the drug, how it's working. So if there is a telltale sign that if you do a superiority trial, it will possibly fail, then you have 2 options. Number 1 option, throw the drug out. It's out of the contention. However, if it is possible that, that the new intervention that you are trying to study has certain desirable features. For example, it could be less invasive. For example, the current drug has to be injected, and this could be as a pill. It could be less expensive. It could be less debilitating. It could be, for example, the new drug could maybe have to be taken only once in a day where the old drug has to be taken every hour. So there could be certain features which makes your new drug not directly superior, but maybe very close to the existing drug. But it may not be able to beat it, but it will be very close to it. And it has certain features which will make it attractive to maybe not all patients, to a subgroup of patients. Then you may want to pose the trial as not a superiority trial, because you'll fail. There's a good chance of you'll be failing. Then you may start with a non-inferiority trial. Now you may ask the question that, okay, I do the trial, non-inferiority trial, and I am successful and declare non-inferiority. And in fact, there is evidence that it is not only non-inferior, it could be superior. Now, can I claim superior after non-inferiority? The answer is yes, you can, with certain cost. So other way around is not true. That means if you do superiority trial, fail it, and then claim non-inferiority, you cannot do that in a single trial, you do a completely new trial. But if you claim non-inferiority, and you are successful in doing that, and there is a further evidence that there is superiority trial, you can do it certain price. Okay, so that's why sometimes it's advantageous when there is no clear evidence of superiority to go non-inferiority trial. Okay, so again the question is that you are talking about intervention which is slightly less efficacious. Now, less efficacious is a vague term. In statistics or in mathematics, you have to give a quantitative notion. What is the less? What is the big? Okay, so NI trials or non-inferiority trials are typically active control trials, as you can see, because you are comparing your intervention against an existing intervention. It's an active control trial, okay? So active control trials are special type of trial, okay? So let's try to see in a picture what we are trying to do. So we have three diagram here. So basically, you have to first declare how much effect you are going to lose, and you are still going to be okay. So let's say delta, which we call some kind of margin, okay? And delta can be taken positive or negative. For this picture, I took delta to be positive. So for a superiority trial, you're trying to declare that, you know, my mean of the current drug or the new drug is better than the existing one. So basically, this is the picture. When you're going to equivalence trial, okay, you can do a toast kind of experiment, like two one-sided test, and you are okay if you are within this plus minus delta region. A lot of bioequivalence trial are like that kind of trial. And non-inferiority in the middle, where you say that, okay, I will allow this much loss of efficacy. Now, there's a challenge there. The challenge is that this delta, if you choose to be too small, then you should do a superiority trial. Why you are bothering about non-inferiority? Now, if you do delta to be too big, then you are going towards placebo, no effect. So your delta has to be somewhere in the middle, and that's where the crux of the problem is. We do not know where to put it. And there is no single formula which declares this delta is good for everything. It declares about the disease, it talks about the disease, it depends upon the outcome, whether binary or continuous, it depends upon many factors. So there are guidelines, but the guidelines are like, 10-year system, like, there is nothing, no rule. Like, it's a vague points. Okay, so there is no rule, there is no formula directly which you can, you'll be fine with. So, choice of margin is a big issue in non-inferiority trial. It's a huge deal. It can actually make or break your trial. Okay, so ICH, the International Committee of Harmonization, then the FDA E9 guidelines, they give some bullet points, but there is no formula, and there is a reason there is no formula, because there is no single formula which works for everything. So, in this particular talk, we will be focusing on non-inferiority trial for a special type of RCT, which is a smart RCT. That kind of thing we're talking about. But let's talk about non-inferiority. Let's see, is it just changing the sign of the hypothesis? That's the price? Or what are we going to pray? So let's see, in classical non-inferiority trial, you don't have a placebo arm. Okay, because that doesn't make sense. It's not ethical. You have an existing intervention which works. So let's compare your new one with the existing one. Okay, so this makes ethical sense, but you have to pay a huge price. So let's try to use some notation. In experimental intervention, I call it E, and then the reference or the active control, which is the existing regime that already works, is called R. Okay. So without loss of generality, we assume that the higher mean means the better efficacy. If it's go other way around, for example, you are trying to do blood pressure reduction, just change the sign. It will still work. Okay. So done mostly for ethical reason, so basically you don't have a placebo arm. So it requires a clinically acceptable margin. So I am now defining my margin as small delta. I will not work with capital delta. I will work with small delta, which is negative quantity. Okay. So you can think of, I have these three kind of triangle kind of diagram. So I have experimental arm, I have a reference arm, and a placebo, which is a putative placebo. That means I don't have a placebo. So these lines are dotted line because I don't have this guy in my current trial. I have two-arm non-inferiority trial. So what do you want to test? You want to test basically whether my mean, mu-e, the mean of experimental minus mean of the reference drug is less than or equal to some delta, and then alternative is that this is not true. Okay, so we will be happy to declare H0 is false, and then we can go for this one. Well, this is two-arm NI trial, and I believe Bhivash already wrote a very nice paper, which is in the final stage, to doing non-inferiority trial, this kind of non-inferiority trial with the smart setup. Okay, so what is the picture? The picture is that, okay, if I can draw this way, this is my reference. Okay, I know where it stands, and mu p is somewhere there. Now, somewhere there, why? Because I do not know where it is, because I don't have it. Okay, so I have a dotted line here. Hopefully, it is there. If mu p is here, I'm in trouble, because then current drug lose many of its value. It happens, you know, like because of some of the drug loses value because we are eating genetically modified food, environment is changing, all this thing. And then what I'm trying to claim that if I take mu r plus delta, remember delta is negative quantity, then if mu e falls in this region, then it is non-inferior. If it is fall this region, it is not non-inferior. It's essentially, we cannot declare non-inferior. Okay, the problem is that you are making an assumption. The assumption you are making is that your mu p is here. You can check it because you don't have a placebo arm. Okay? Okay. So, since in a two-arm non-inferiority trial, your placebo arm is absent, you are making some implicit assumption. You make many more assumptions. I'm just putting 3 on the board because they are the most important one. One is constancy. That means the historical difference between the R and P are still valid, because if it is not still valid, I do not know where P is right now. Okay, so that's the price. Okay, you are paying because you don't have a placebo arm. Okay, and assay sensitivity, which is much more general thing, the ability of the current trial to distinguish an effective treatment from a less effective one. So in a two-arm NI trial, which is a much more popular non-inferiority trial, we cannot do anything without external validation. Okay, that means the information about the placebo actually comes from history. You have to depend upon history when R got established. Okay, of course, when R got established, we are assuming that there is no other drug, so R must have been tested against a placebo or standard of care, and it's a winner, right? So you have to have that information, and you should be using that, assuming that all this is still valid. Maybe 10 years back it was produced. It's a huge assumption. Okay. Okay, so to avoid some of this assumption, and in fact, I'll tell you that European Medical Agency was much more flexible than the FDA. So a lot of the initial work about this 3-arm NI trial we'll be talking now is actually came from Europe. You know, a lot of European scientists, they published the initial work on 3-arm NI trial. So what they did, they say that okay, if there is no big ethical concern, you can have a placebo arm. So where we are going? So in this triangle, now we are telling that, okay, I have E, I have R, and I have P. And I have drawn 2 solid lines. Okay? You can also test E versus P, but that is redundant. You don't need to do that testing, because if you test E versus R and R versus P, that is kind of automatically can be checked. So what they did, so resulting design is a 3-arm trial, which they called gold standard design. There is no gold, that's the name is the gold standard design, okay? So which does not require external validation, because you do know now where the placebo is from the current trial. So now this is the picture I get, okay? So mu r is here, mu p is hopefully here, but we know where it is. If it is not here, if it is here, then R has no value, you can throw it out, okay? But you do know, there is no assumption. And then you declare some delta, okay, and you are still declaring non-inferiority if mu experimental falls in the middle. Again, the question is that you gain something, but you still haven't solved the whole problem. Why? Because you have to still talk about delta. Okay, you kind of minimize one assumption, because I know where the placebo is, okay? But still you have to talk about delta. And again, like, you know, even for 3-arm trial, we have a little bit more information, but the choice of delta is still not clear, okay? So the first paper, again, as I told you, most of the initial papers came out of European statisticians because it was being allowed there. I should also warn you that 3-arm NI trial cannot be done in every disease. If it is cancer, you cannot do 3-arm NI trial, okay? possibly in those areas where the disease is not mortal, like for example chronic disease, for example, you can do 3-arm trial. But this is not a thing that can be applied everywhere. Just like SMART cannot be done everywhere, this cannot be done everywhere. Okay, but there are situations, for example, weight loss program, the skin disease thing, and there are other chronic disease area where having a putative placebo is not bad. So again, we have to choose some delta. Okay, now this choice of delta, it has diversified the research of non-inferiority into many different directions. Okay, I'll choose one, but I'll show you another direction that is also possible. We haven't explored that in this particular talk. So Piaget came up with an approach. What is that approach? So what Piaget cleverly declared that Piaget said that, okay, I will choose my margin as the observed difference between the current trial's reference mean and the placebo mean. Okay, so what does that mean? So I do know the difference mu r minus pup. I don't have any external validation or from history. I will take this whole difference and then choose a fraction of the difference as my margin. Okay, so you are not declaring my margin as like 50%, like 0.5 or 0.7 or 0.9, like absolutely, but you're declaring that how much I want to retain. Okay, that's what you call fraction margin approach. So what does it do? So you kind of declare your margin as a function. Now everything is from current trial, which is good, you know. So you can see, so what he did essentially, instead of delta, he put the whole thing like mu r minus mu p times some sigma, which is here. Okay. Now if you do some algebra, not a big deal, you can write down the whole thing in terms of linear contrast. Okay. Now you see this thing makes a lot of sense. Why? If you put theta equal to 0, then you see that mu r vanishes completely. So you are essentially testing mu e against mu p. That means r is no use. Throw it out. Okay. If you put theta equal to 1, then mu p is in the garbage. You don't need to consider placebo at all. You can just test mu e and mu r. So theta is between 0 and 1, and it's your belief how much you want to retain. Okay. It controls everything. That's why you call fraction margin approach. Well, it works well, but essentially every approach, every model makes an assumption. This model also makes an assumption. The assumption it makes that the only time you can construct a non-inferiority margin provided this quantity is positive. Now, what if mu r minus mu p is almost on the top of each other, or what if, like, you know, drug, the current established drug lost all its efficacy? It's possible, you know, you never know. So this approach makes assumption, though it's very unlikely to happen, but you are making the assumption that mu r minus mu p is strictly positive to make this claim. Okay, so Piaget essentially argued that one first must reject the alternate AS or the sensitivity hypothesis. So essentially what he is doing, we are testing E versus R, but we are assuming that R versus P is kind of a gatekeeping, you know. I'll just observe R is better than P, and once R is better than P, I can just do and test E versus R. That's what he was doing, okay. So he is not testing R versus P, but using as a gatekeeping condition, okay. So due to the hierarchical nature of K naught and H naught, K is for AS as sensitivity, and H is for non-inferiority. You don't need to do any alpha adjustment because you know there could be multiple testing issues, so you are not doing that. They provided power as a function of this quantity, the fraction margin. So essentially you are not specifying directly a quantity which you have to do in a 2-arm trial, but you are trying to just telling that how much percentage of the whole effect I want to retain. You may call 90% effect, you may call 80% effect. If you call 100%, that's basically superior trial. Okay. So Koch and Tangent into 1998 suggested the difference between R and E is expected to be much smaller than the difference of the both treatment compared to the placebo. So it essentially points out to the fact that non-inferiority trial is an area where you should not probably go with equal allocation. We know that, you know, for if I have a two-arm parallel design, I get maximum power if I do 1:1 allocation. But in fact, non-inferiority is an area where actually it can be proven that, you know, if you do equal allocation, your power will not be optimal. You should have a smaller placebo arm. Actually, it's almost can be guaranteed. So that's the thing I talked about. Equal allocation may not be optimal for NI. Again, it's a different question, like how do you find out optimal allocation ratio? We haven't shown that here, but can be done. There are papers on that topic also. Okay. Now, again, as I told you, every approach is as good as the assumptions like if assumptions holds. So there is a competitive approach, and I'll just touch it, and I have several papers on that particular approach, not for the SMART, but on the non-uniformity context. So Hida and Tango suggested that, why to make assumption? What if you do test H0 and K0 together? That means I want to test and claim that my reference drug is still better than the placebo, and I want to test and claim my new drug, new intervention is as good as the reference drug. I want to claim the both. You can do that because you do have all three arms, okay? So this approach is known as fixed margin approach. The challenge with this approach is that it's essentially a continuation of two-arm NI trial because you are not constructing your delta because you are testing both. To test both, you have to supply the delta just like what you have done for two-arm trial. So there is no free lunch. So each way you have to pay a price, okay? Note also, like essentially to do this kind of testing that Hida and Tango suggested, you will have to jointly reject H0, K0 together. Now that is very, what can I say, very conservative approach. We can prove that actually. So this is based on intersection union testing principle. No alpha adjustment is required. However, it can produce a bias test. There is a very nice paper, not from clinical trial, but from Technometrics by Berger, that if you do this kind of all or none procedure, you don't need alpha adjustment, but you could end up with a very bad test, bias test basically, okay? So in this particular talk, we'll talk about the fraction margin approach. Now these things can be cured, by the way. But like for example, can you cure Hida Tango's approach to not to produce bias test? The answer is yes, you can. You have to do some mathematical juggling, and I have done paper not on SMART, but simply NI trial, like that can be cured, but I'm not talking about that here. In fact, we are talking over the lunch that what can be done, this can be done. And you can compare like, how fraction margin and fixed margin approach compare their performance in the long run, but I'm not talking about that here. So again, so we'll be talking about the fraction margin approach based on three-arm SMART. Okay. So let's now talk about the SMART design. Again, this is where I took Bivash's help because I'm not a SMART trial guy, but I'll just tell a little bit about what is adaptive intervention, what is SMART. So an adaptive intervention is essentially a sequence of intervention and which is adapted depending upon how a particular individual is performing given a particular intervention, and essentially it is also named as dynamic treatment strategy, stepped care. They are all different kind of name people have used over the years. They all mean pretty much same thing. And SMART is experimental design to test adaptive intervention. So it's a novel trial design that was developed to inform the developer adaptive intervention. It's a multi-stage trial. So essentially, adaptive intervention is not same as adaptive trial, as we all know. So each stage corresponds to a decision rule. At each stage, subjects are randomized to a set of treatment options. So essentially, it informed the developments of AI and develop a proposal for an adaptive intervention, which could then be tested in a two-arm randomized trial. The typical SMART are two-arm, to begin with two-arm, and then they bifurcate into more than two-arm. So again, SMART comes in different flavor. For example, we will be talking about a special type of SMART here, okay? So again, this is a standard SMART. Again, as I told you, for example, in the initial stage, you randomize into either A or B, and then if A, then you have to have a outcome variable, okay, proximal outcome, depending upon whether somebody is a responder or not. You may re-randomize it again, or you may just keep it. Now there are other kind of SMART where you randomize both. Actually, that also exists. So we didn't consider that. We particularly considered this particular SMART because our example is actually supporting this kind of SMART design. So again, I denote by this R means randomization. So first interventions are A and B. In standard SMART, the placebo arm is absent. You don't have a placebo or a control arm. So both arms are intervention arm, which makes ethical sense. Based on the individual's progress during the first intervention stage, if he is a responder or non-responder, which need to be defined a priori. Somebody is a responder, you continue the same treatment strategy. If somebody is non-responder, you may want to augment it. Depending upon, everything has to be protocolized and has to be determined a priori. So at second stage, so that's what I told here, responders are allowed to continue, and I denote them either by V or M, or if they continue, then continue with the existing intervention. So treatment alternatives like must be used or could be used in a practice. One may ask a clinician or a community organization, for example, how to determine that. You have to determine a tailoring variable. So SMART is not so easy. You are asking quite a bit a priori from the clinician or the doctors like whom you are working with. And you also should have the ability to collect research evaluation on a longitudinal basis. Okay. And SMART design may differ depending upon the ethical feasibility or the other concern. And our goal is to keep SMART simple, but again, as you can see, there is a lot more. It's quite a bit taxing depending upon like what you want to do. So let's say an example like which motivate us. So now this is an ongoing example, like it's an NIH-funded R01 study for cancer symptom management. So basically, this is the protocol that you see here. Essentially, the sample size that they calculate. It's still in the field, so they're still collecting sample. So we have 331 subject, and we have 2 lines of treatment, reflexology and the mediative practice to essentially to manage the symptom. And then this is kind of novel. There is a control arm, which is not seen in standard SMART. Okay. Now this has been put because of some reason. Like first of all, NIH wanted it, and there is other reason that they have to put a control arm. As you can see, they are not 1:1 allocation. They are like 2:1:2 kind of allocation. Okay. And again, as you can see, if you responder, it's being continued. If you non-responder, then there's some augmentation going on. Okay. This is our motivational study. So okay. So let's try to see them side by side. So control arm is to compare it with the intervention sequence. Control arm is recommended because initially when they submitted the R01, they don't have a control arm. They have standard SMART, but NIH wanted that arm. So this is standard SMART, and this is SMART with a placebo or control. So as you can see, if you're in placebo arm, there is no more randomization. Only one time you randomize to either A, P, or B, and then you continue. Okay. So okay. So let's now stick to some notation. They are not very different. They are changed a little bit to accommodate the placebo arm, basically. Okay. So depending upon somebody's responder or non-responder, you may have 5 adaptive intervention. We call it D1, D2, D3, D4, and D5. As you can see, D5 there is no re-randomization. There is no responder status judgment. And then you can have a distinct path, or you can have a shared path. So it's distinct path starting with different initial intervention, and shared path starting with the same initial intervention. So T1 and T2 are denoting the intervention option at stage 1 and 2. R is the responder indicator, which is either 1 or 0. Remember, for the placebo arm, there is no responder determination. So observed data is essentially for ith individual consists of this 4 quadruple, which is like T, R, then T, and Y. Y is the final outcome. So an individual's potential outcome in the Robbins framework is Y, T1, T2, where T1 could be either A, P, or B. That means you can either be in the initial randomization, either could be in the arm A, placebo, or B, or it could be T2, which is A, V, and M. Or it could be, if T1 equal to A, or it could be T2 is equal to this thing if T2 equal to T1 equal to B. If T1 equal to P, it doesn't change because there is no other randomizations going on. So we also assume that our mean is defined like that and the variance is defined like that, just like the standard SMART. So again, some more notation. Again, it's very similar to the SMART except for the fact that we have to always accommodate the placebo arm. That's a change we're trying to do. So pi T1 is the first stage randomization probability, and pi T1, T2 is the second stage randomization probability. As you can see, typically if I have pi T1, then pi T2 is 1 minus pi T1. But since I have 3 arm here, so my pi P, the placebo arm probability, is 1 minus pi A minus pi B. That's the only change we have to do. And then these things are standard like that we do. A responder is assigned to the treatment sequence T1, T1 with probability pi T1 because there is no change. And then a non-responder is assigned to the treatment T1, T2 with this joint probability. Using the principles of inverse probability weighting, which is very standard into the SMART literature, you can actually weight the ith individuals embedded attribution like this one. So now we can define the observed mean, which is nothing but a weighted mean because you're getting contribution from different arms. So we can define gamma T1 as the response rate for the initial intervention option for T1. So and with respect to some pre-specified definition of responder. In this case, P pi P equal to 1 because there is no determination of response status. Okay. So we can determine the population mean, which is nothing but the weighted mean. And we can also calculate the variance. They are a little bit like tedious calculation, but nothing special, very similar to the standard SMART. For the shared path, the covariance between the YD1 and YD2 can be also determined by standard calculation. So let's talk about the example. Okay. So existing method for SMART study is to compare adaptive intervention and associated power planning resources and are suitable for traditional superiority testing. But here we are talking about non-inferiority testing. So in NI testing, the goal is to establish that a new intervention is almost as good as the existing one with certain delta or loss of efficacy. Okay. So we will be talking about delta in terms of percentage, like how much you want to think. In the 3-arm SMART study, the goal is to test that whether a new intervention is non-inferior to the existing one. And we are talking about intervention means adaptive AI here. Okay. So before combining the idea of non-inferiority and SMART, let's revisit the example. So again, this is the example that we have. Note that this particular study, when planned, is actually not planned as a non-inferiority trial. Okay, so we are just giving an example to you that okay, this kind of SMART can actually happen in the real world. So the participant flow through SMART MR. MR means the mediative practice or reflexology for symptom management during the cancer treatment. Counts are what we projected before the study. Essentially, the numbers that we have shown, like about 150, 50, and 150. The projected sample size, they're still collecting sample at this point of time. The interventions are reflexology and mediative practice and standard of care, okay, or the control. So in SMART MR, we have a sequence that starts with reflexology, then mediative practice, intervention is added if required. Or it starts with the mediative practice and then reflexology is added. And in a control arm, essentially you don't do anything. You just do leaflets and stuff like that, okay. So also in SMART of self-management intervention, one cannot switch to another intervention. That's also a restriction for this particular example. So let's talk about NI with SMART. Let's try to see like how can I manage or marry both the ideas. Okay. So again, this is the picture, the thematic picture. So I have A, the intervention A, and then P placebo or control, and the B, the second intervention 2, and then this is the thing. So we have a reference which is we call DR, experiment we call DE. So essentially we are using same kind of notation. And the placebo or the control we call DP. So, and corresponding means are denoted as mu of each of these arms. So, in terms of SMART design, essentially I have to declare a margin, remember delta, okay. So, I declare my delta again, just like a function between the observed difference between the reference and the placebo. Very similar thing that we have done for 3-arm NI, except for the fact that I replace the mean, now the adaptive intervention or interventions mean here. That's the change. Nothing else has changed. So again, I have 3-arm DE, DR, and DP. Okay, and then I can write down the NI SMART very similar fashion, just like what we have written down for standard NI trial, except for the fact that now I have adaptive interventions mean. So we illustrate NI testing for distinct and shared path. So distinct path, as we have mentioned, in distinct path is starting with the different initial value. So basically, let's compare D1, D5, and D3. So we can write down them as in this particular fashion, and you can actually calculate that this is your test statistics, and you can also calculate standard deviation. Once you calculate the standard deviation mean, you can do a large sample test, which is nothing but a normal distribution. And then once you have normal distribution, you can also calculate the p-value or the rejection region, and you can also get a sample size based on this particular formula. Nothing special going on, except for the fact that you are plugging in the standard mean by means of the adaptive interventions mean for that particular path. So, and this is the standardized effect size, which is the Cohen's effect size we have used to calculate sample size and power. For shared path, essentially we have taken an example, D1, D2, and D5. So in shared path, essentially starting with the same initial intervention, you can also go to the other side and it will tell you the very similar story. So again, this is my test statistics. You can calculate the variance. Once you calculate the variance, you can again use a large sample approximation for based on normal distribution, and you can again calculate a sample size by using the formula here. Okay, nothing special. Everything is standard calculation. So let's see some simulation studies for the 3-arm NI SMART to explore its power. Again, this is more a design paper, not so much about analysis paper, like to see like how do you do sample size and power. So in the simulation study, everything is kind of pre-specified. You kind of pre-specify what is standardized effect size, you essentially, this particular way we compute the power is not actually based on the normal distributions formula. It's a Monte Carlo power that we calculate here, and we try to see how good the Monte Carlo power compared to the normal distribution based approximation based power. So as you can see, we are essentially counting like how many times we are rejecting the H naught. So you can use this particular simulation to detect both type 1 error and the power. So here is the table. For example, this is the table for distinct path, and as you can see, we have standardized effect size. This is also a function of responder rate for each arm. We assume that, to simplify the life, that we assume that responder rates are equal, but if you have differential responder rate, the table will be a little bit more complicated, but that can be done. So as you can see, like, you know, it typically requires somewhere around like 170 to 200 subject, depending upon the standardized effect size. I should also mention that non-inferiority trials are typically larger trials than superiority trials, because the difference between the two active intervention, the existing one and the old one, is typically much smaller. So typically we expect a larger sample size. That's why it's not so easy to implement the non-inferiority trial. If I go to shared path, this is the picture for shared path. Shared path is a little easier because of the homogeneity of the variance. The sample size requirement is little less stringent, as you can see here. So we have done that also. Again, we may make a lot of assumptions to make this table, but again, like, the simulation setup is such that if you specify me the differential responder rate, I can always calculate the sample size. One thing that we have not done in this particular work is that, for example, we are talking that, you know, what if I want to calculate the optimal allocation ratio? So in this particular example, everything is the 1:1:1, but that can be done too. In fact, I have paper, not in smart context, but standard non-inferiority context. Like, I want to calculate the NE, NR, and NP as a proportion of inverse of variance. I can do that. That can be done here too. The other question that we also discussed, I think, over the lunch, that, for example, I think somebody asked, what if I want to calculate the optimal probability of allocation? We haven't done that. That's a hard problem, okay? So this is some more picture. So this is the power curve for different values of sample size. Now what we have done, we want to retain 80% effect size. Now if we want to retain 90%, we get very similar looking curve. So this is the power thing, and for different values of n, but you want to retain 80% all the time for distinct and shared path. Then I have another picture that is the power curve when n is fixed, but I vary my theta. Like, for example, 0.6 to 0.9. Note that you don't want anything below 50%. Then non-inferiority doesn't make sense. Okay, it have to retain at least 50%. That's the rule of thumb. We also did some more picture for example, we are interested in heterogeneity of effect size. Like, for example, if one arm is very different than the other arm, we have done that too for different values of sigma e. And as you can see, this is the picture for power and for different values of n. Now future work. Now lot to do here. I'll just present you some of them because we are just scratching the surface here. Okay. So joint testing, as I told you, for which proposed by Hidayah and Tango, if we cure the too broad strategy of testing AUT principle, we can actually do joint testing principle of Hidayah and Tango too. Sequential testing, that's the what can I say? That's the state of the art in non-inferiority testing. What a sequential testing does, you essentially test AS first as a gatekeeper, and then see how much you have spent, and then use the remaining to test your NI. Actually, we just finished writing a paper on sequential testing, and we can show sequential testing has better power than both fraction margin and the goal and the both thing. Not for the smart context, but can be done here too. So in this study, we have explored continuous outcome. Now I think Polash has worked something on the binary outcome, but when you go to the binary outcome, things will be even more interesting for non-inferiority trial. Why? Because the delta margin, the way we are constructing it, the difference of mu r minus mu p, that's not unique as soon as you move from binary outcome. Because your binary outcome depends upon the type of the functional. Are you testing relative risk? Are you testing log odds ratio? Are you testing the relative risk difference? Every time you change the functional, your risk margin construction method will change. We just published 2 articles. One is in biopharmaceutical statistics and one in the CSDA to show that for non-continuous data, for example, survival data, for example, binary data, Poisson data, your margin construction is not unique. And in fact, if you are a non-inferiority researcher, FDA came out with guidelines for doing non-inferiority testing, I think last year. And Ram Tiwari, who is my collaborator for a long time, and he's the director of FDA essentially. So we actually showed that particular guideline has a huge hole. Because it talks about unique margin, and unique margin exists with the current FDA guidelines, provided your outcome is continuous. If your outcome is binary or non-continuous, FDA needs more guideline to make things continuous. But I'm not talking about that one here. So that is a problem here also, because you're testing non-inferiority here. The other thing that is interesting to me, that in any active control trial, what is active control trial? You're talking about existing drug. And you're talking about existing drug means you have historical information. So apart from using, where the information is used here? Nowhere. It's not used. So in 2-arm trial, you only use historical information to construct the delta. If you take a Bayesian strategy, you can use actually a lot more. And in active control trial, Bayesian strategies are kind of standard. Because you have information about the placebo, you have information about the existing drug, you may or may not have information about the new drug. So Bayesian works come naturally into the non-inferiority trial. Like whether you are a classical or a Bayesian, you are using the prior data some sense or other. So doing Bayesian smart, even you are not talking about non-NI, is something like, somebody should take up. Okay? So, and especially in the Bayesian NI smart is something that should exist, naturally. So similar method can be done for NI smart framework too. So I'll stop here, and if you have any question, I'll take one. Thank you. Mm-hmm. Example.
Yeah, example. And then it's clear which is active control, which is the new treatment, which is control, right? So if you have more than 3 off.
In the smart?
Yeah, in the smart. How you determine which is the active control,
So again, I'm pretty naive here. So do you have smart where you have like 1, 2, 3, 4, something like that? Does it exist? So basically talking about initial thing, then you have 1, 2, 3, 4, something like that, right? So I can think one example. I can tell you one example, not in the smart context. I can use the board, right? I can use the. No. Can I use the board here? I can write here. So one example I can give you, not in the smart context. Let's say I have this kind of thing. So this is my control or placebo, and this is my dose. Dose 1, dose 2, dose 3, dose 4. Okay. You want to see at which particular dose I am in the boundary of non-inferiority. That has been done. I have done some work on that, but on the dose aspect, not for the. So you can think of similar like treatment 1, treatment 2, treatment 3, treatment 4. It will be interesting to do in the smart context, because then you have to bring in those probabilities and things there. Yeah. If you have an example, sure. Sure. But that kind of thing I have done, like we call it like multi-arms NI trial. We have done it. Because as you increase the dose, you increase toxicity too, right? You don't want to, so you want to end as soon as you hit the boundary. That I have done, but yeah. It's a different context, right? That makes more sense, yeah. No. Typically, people either minimize the variance, total variance. That's one strategy. They sometimes minimize the cost. So, like, you have to have some kind of utility function, some kind of utility, like, total variance, total cost, something like that, or can be a combination of that. So people have done that in the standard NI context. I'm sure can be done in the smart context. Like for example, you have multiple apps or multiple mobile health, and you can declare, for example, not just the cost. For example, you can declare like what's the expenditure, like how much. For example, one app may require 5-minute attention, other app may require a lot more. So you can declare something like so you can do something like that also. Yeah, but it is pretty much proved that you should not do 1:1:1.
So let's say three answers, the outcome three answers the same variance. That's the one.
Yes. So all this power calculation I have seen, except for one picture, we assume that it's homogeneous. But one graph, actually, Vivash came up with the idea, what if the things are not homogeneous, right? So what I did here, I assume, like, let's say variance is 1:2:1. So yeah, but that will determine, like, that don't have to be equal. Typically, they're not equal. Absolutely. Thank you.